systematics and this introduction to grading plans. We only have one topic left we're talking about for this PowerPoint. I have posted this in Canvas already, so if you have a computer and you want to pull it up today, you're welcome to do that. We're basically talking about a characteristic that is found in all land plants, but because it is not exclusively found in land plants, there are some types of green algae that have this as well. We can't classify it as a shared drive trait. Because a shared drive trait is a defining feature, something that's found only in that group and no other group. But because they all have it, it's kind of an important feature to talk about because all plants are going to be multicellular, and this really does relate to that. So here's where you can see this appears in evolutionary history, so you can see there are some lineages of green algae here that have this characteristic, but it's also found in all of our land plants. Plasmodesmata, we talked about, are essentially channels or tunnels between the cell walls of two cells that connect each other. So you've got a tunnel in this cell wall, this cell, and you've got a tunnel in the cell wall, this one, and it goes straight through both of them in the same position, the same spot, so that they can essentially communicate more efficiently with each other, right? You've got interactions between their cytoplasms, their hormones are going to be able to communicate, which is, those are communication molecules, and even in some cases, the organelles will actually traverse into the other cell. And this is going to be something that they use for the transfer of compounds. If you think about how compounds normally have to enter a cell, usually there's this kind of filtration process where you have to come out of the cell first through a semi-permeable membrane, undergoes kind of filtration, let some things out, some things not out, go through that process, go through the cell wall, then it has to make its way back into the next cell next to it, so it has to go through that cell wall and the semi-permeable membrane of that one. That's a slow process, right? This is much more efficient because they're directly connecting to each other. And if you think about it, if this compound is safe to be in this cell, it's probably safe to go into this cell as well, so you may not need to go through that filtration system. And so this is adaptive for multicellularity. And let's talk about why it is. Usually when I've got some sort of a shared drive trait or feature like this that I'm talking about in class, I'm usually going to discuss why it's adaptive as well. So for example, when we get into land plants, which we're going to get into next, we'll talk about the shared drive traits for them and how that relates to their making a transition from an aquatic environment to land, and why it's adaptive for being on land. Okay? So let's talk about multicellular organisms. In multicellular organisms, you've got many different cells, and typically those cells are specialized for different functions. I'll actually use humans as an example. So you have skin cells, you have nerve cells, you have red blood cells, you have white blood cells, you have liver cells. They all do different things. They have different jobs. Right? Again, it kind of works the same way with organelles and cells. When you have something big like this, a body big like this, you compartmentalize the jobs. Different things, different jobs. Even plants are going to have this. They've got root cells, they've got stem cells, they've got leaf cells. They all do different things. And when you have this sort of situation where you have these multicellular organisms and specialized cells, they work together and interact with each other to form higher levels in the organization hierarchy of life. So cells are grouped together, work in a coordinated fashion to make tissues. Those will coordinate together with each other and form into organs, and then you have organ systems and so on and so forth. So that's going to apply here to the plant. But to form tissues, you can't have just the cells next to each other and just sitting there. They do have to communicate with each other. They need to be working as one entity. And so how do they do this? Well, they physically stick together and form the tissues, but then they have to communicate, and they do that through chemical interactions. So that involves chemicals. Right? These molecules that we were talking about that are communication molecules. You have to send this information from one cell to another. I'm doing this, you need to be doing this too. Okay, so how do I communicate that with chemicals? That's how I'm going to let you know what you need to be doing. So you want this to be efficient. If you have a big organism that's multicellular, you want to be sure that they can communicate efficiently and quickly so that they can function. Okay, well, that's what these plasmodesmata do. They enable cell-to-cell communication and coordination of function. It's a much more efficient way to transfer nutrients, a much faster way as well than to have to go back through the cell wall, cell membrane, and back into the other cell through their cell wall, cell membrane, and have to keep doing that to go from one cell to another. That's incredibly slow, and it's inefficient. This way we can connect them directly, and they can move that from one cell to the other essentially immediately. And that is going to unify most of the organism into essentially one living continuum. That's what we need a multicellular organism to do. Their cells need to be working together so that they are working as a whole, right? Not as individual cells. Okay, now at this line, this is where you can draw a big line in your notes. This is where exam 1 ends. Okay, so this is what you'll have for Monday's exam. What we're going to move into now, which is essentially going to one of the groups that are in these green plants we've been talking about, the land plants. We're going to talk about that for exam number 2. This will be exam 2 material. Okay, so when I say the word plants to you, I am actually referring to this clade right here, land plants. When people usually talk about and they say plants, this is what they're referring to. I know that the previous group, the Florobioida, the Virid plantae that we just discussed, was referred to as green plants, but it technically was more than just plants. It had protists in it as well, specifically green algae. When we say plants, this is what we mean, the land plants. So this is the primary focus of this particular course, because we don't really deal with green algae. Land plants are going to be clade inside the clade that we've just been discussing. We've been discussing the Virid plantae, which includes our green algae, which are right here, that's a paraphyletic group, and this monophyletic group right here, which are called the embryophytes. We'll talk about why land plants are called embryophytes. And so this PowerPoint is going to be all about those and what their shared drive traits are going to be. You'll notice I keep giving you a list of shared drive traits. You're going to need a lot of those. And now's the time to start practicing keeping them straight. Because you'll definitely, when you get to the final, have multiple lists that you're going to have to be able to match with the right type of group or clade that we're going to discuss. So start practicing writing it out, okay? So write green plants on a sheet of paper, put a line over underneath it, list out the shared drive traits, okay? Next to that, put land plants, put a line underneath it, start listing out the shared drive traits. Make sure you know which goes with which. So for example, later when we're talking about flowering plants, seed plants, things like that, if you tell me that seed plants, one of their shared drive traits is flowers, it's wrong. That's a shared drive trait for angiosperms only. That is a group of seed plants, but not the only group of seed plants. So it gets more complicated to keep them straight the more lists we add. So that's why I say start now, so that you can start getting familiar with which ones go with which clade. Okay? This is a monophyletic group that's within our green plants. And if we go to where it is in the cladogram, right here, and follow the line down, you'll see this grouping of lines right here are the ones that are closest to that branch of green algae, right here, which are what are referred to as the charophytes. Okay? This is the only present-day algae group that actually shares certain distinctive traits with the land plants that suggest that they're related to each other. Okay? So if you look in charophytes, this group right here, this group of algae right here, you'll find that their cells have circular rings of protein that are embedded in their plasma membrane, as do land plants. Their job is to essentially make the cellulose in the cell wall. And then if you look at the structure of their sperm, it's going to be a flagellated sperm, which means it has a tail on it. Right? And it is similar to the flagellated sperm that we find in some of the land plants. I say some of the land plants because some of the land plants, the more evolved ones, have actually evolved to lose that tail on their sperm because they don't need swimming sperm anymore. They use pollen to transfer their sperm. But their ancestral sperm did have flagella. Okay? So remember, you can have derived traits that are lost over time in evolutionary history in replace with new traits later on. But for the ones that still have that flagellated sperm does look like the flagellated sperm that we see in these charophytes. Okay? So it suggests that these are the closest living relatives of plants, not their ancestor, because these are living modern day lineages, okay, but the closest living relatives of the plants. Okay? That's also supported by a lot of other types of data, including molecular data. Okay? So we've talked about how important molecular data is for kind of confirming some of the things that we come up with in cladograms, hypotheses. We come up with cladograms based on morphological characteristics. These are morphological. These things right here, morphological, if you can support that also with molecular data, that provides a lot more support for your hypothesis. Okay, so what? These are still green algae, so why do we care that these are the closest living relatives? Well, that means that they can tell us something. They can tell us a little bit about what that ancestor of the land plants might have looked like. Okay? As you can see here, the branch for land plants branches off from this group right here. Okay, so how similar looking was it to these charophytes, also known as stoneworts? One of the most important things that we can tell from this is that ancestor was most likely aquatic, because that's what green algae are. Green algae are aquatic organisms. Now, based on the common name of the clade we're talking about right now, land plants, that tells us there's a big change that had to happen here. Now, that being said, not all land plants live exclusively on the land. Okay? There are a few species that have returned to aquatic habitats over their evolutionary history. For example, water lilies, right? Water lotus, things that you're going to find in Reelfoot Lake. Those are in fact land plants, even though they live in the water. Okay? They're not things that have never made that transition to land. In reality, what happened is they made the transition to land, okay, which means they're going to have the features that land plants have that make them adapted for living on land, and then they have gone back to the water and now have new drug traits to help them deal with going back into the water. Okay? So there are some things that live in the water now that are still classified as land plants. Most present day land plants, though, live on land. And that's going to present some challenges if you used to live in the water. Okay, so one of the things that hopefully you remember from 130 is what the definition of an adaptation is. Okay? An adaptation is some sort of a trait, characteristic that you have that makes you more successful at surviving and reproducing in a particular environment. That last part being important, in a particular environment. Right? So what works well in one environment, probably not going to work real well in a different environment. Particularly if you're looking at different enough environments like aquatic versus terrestrial. Okay? And they are very different environments. Okay? So to make this move to land, if you're something that's adapted to living in the water, you're going to have some changes that happen before you can be successful in a terrestrial environment. Different adaptations are going to be beneficial. Many of the challenges that we see on land are things that aquatic organisms don't have to deal with. And so one of the things we're going to do first is do kind of a comparison of the two types of environments and what's different. Bless you.
Thank you. Bless you. Thank you. Thank you.
As the plants are starting to reproduce. All right. So how is water different than air? Well, for one thing, it's denser. It provides some support to an organism that lives in it. So think about a green algae, especially those ones that are really filamentous, that look like little hairs. If you throw that in the water, you're going to be able to see all of the individual little strands as they kind of fan out and spread out, because they're supported by that dense water. But if you try to take that algae, grab it, and pull it out of the water, this is what it's going to look like. It's going to collapse on itself, because the cells don't have the structural support that they need to stand upright or to fight against the force of gravity, and for that matter as well. So we don't have that support if we're going to be on land that we have when we have in the water. Second, where's all the things that this organism needs? Right? They're going to need nutrients, they're going to need water, they're going to need gases, potentially to do things like photosynthesis and cellular respiration. And in an aquatic environment, that's dissolved in the water. Right? And that water completely surrounds the whole organism. This is an aquatic thing that actually lives submerged in the water. They have water all around them. So if there's nutrients in there, that's dissolved in all of the water that's around them. It's not separated into different places, which is going to be on terrestrial environments. This is a big one though. Reproduction becomes much more challenging if you're out of the water than if you're in the water. One of those main reasons being if you have a sperm that swims, okay? So if you have a sperm that swims and you live in the water, it's pretty easy to do sexual reproduction. You just release the sperm out in the water and they swim where they need to go. They need to swim to the egg at that point. How do you do that on land? And a lot of the adaptations we're gonna see in this transition from an aquatic environment to land environment are specifically gonna relate to reproduction. And the challenges of that, because you have to be able to reproduce. All living things reproduce is one of the properties of life. Alright, now take a look at land. On land, your air doesn't provide that same support, it's not as dense. And so if you wanna grow upright, like most of our land plants do, then you have to be able to support that weight. You have to have some structural support to deal with that. Two, your gases, your water nutrients are separated from each other. Your gases are in one place, the water nutrients are in another place. Okay, so the water nutrients are in the soil, up in the air, and then your gases are up here in the air, but not in the soil. Right? Yes, there are mechanisms to get it in there, but it's a slow process to get that movement to occur. So we're gonna have to deal with that, which means we're gonna have to be efficient at transporting things. Because all parts of the plant need water nutrients, and all parts of the plants are gonna need things like sugars from photosynthesis, which are what the gases are needed for. Here's another challenge to think about. Right? Your cells and tissues can dry up if you live on land. When you're in the water, submerged and surrounded by water, you don't have to worry about that. And cells can get too dry. If they get too dry, they die. So you're gonna have to have ways to protect them from that. Particularly if you're not gonna have access to water for an extended period of time. And then reproduction. By swapping DNA, it's more difficult. There's no water to send those gametes out into. Okay, so let's look at our overview of the land plant phylogeny, or the hypothesized evolutionary relationships between these plants and what we've been discussing so far. There are some shared derived traits associated with them, okay? Things that are found only in these land plants and no other groups, and that defines them. You can see the tick marks are listed right here. So we had a list of shared derived traits for our green plants right here, that was these four. Now we've got another list right here that's going to be specific to the land plants. We'll list them first and we'll talk our way through them. One of them is their life cycle, that they're gonna have two stages in it that the haplontic life cycle does not have, which is the one we talked about that many of the green algae have. It's actually where they get their name from as well. Number two, they're gonna have a type of layering on the above ground parts of the plant that are gonna protect it from water loss. Number three, they're gonna have a special type of cell that we didn't see in any of the green algae. Very versatile one that can do a lot of different things. We're gonna have several cell types that we're gonna learn in this class, and we're also gonna have to keep those straight. And not all of them are shared derived traits for the same clade. Okay, some of them will evolve later on, and we'll find them later on. But they all do different things. They all have different characteristics. They're gonna be different shapes, and they're gonna have different jobs. Some of them will have only primary cell walls. Some of them will have both primary and secondary cell walls. Some of them are alive at maturity. Some are dead. Some are like semi-alive. A lot of different things that are different about them, and you want to keep those characteristics straight. So we'll focus on just one for this lecture first. Then we have some special organs that are going to be responsible for making the gametes, that are going to protect them from drying out and from mechanical damage. So we have our male one, which is the antheridium, which hopefully you remember from lab, and we have our female one, which is the archegonium. These are going to make the gametes. Okay, so let's go into our first one, our first shared derived trait here first, the alternation of generations life cycle. We're going to spend a good bit of time on this because I definitely want you to know this life cycle. It's the life cycle for all plants. So that's the nice thing about it. We can learn one life cycle and we've got it for all the plants. All we have to pay attention to then is which stage they spend most of their time in. Now, this alternation of generations life cycle that land plants have is also sometimes called the haplodiplontic life cycle. The one I gave you before was the haplontic life cycle because the adult phase was a multicellular haploid stage. That was what they spent most of their time as, and they only had a unicellular diploid stage, that was it. But this one has two multicellular stages that they alternate between. One of them is haploid and one of them is diploid, so hence the haplodiplontic life cycle to indicate that we've got two multicellular stages. Remember that they have names related to each of these stages. The multicellular haploid stage is the gametophyte, and the multicellular diploid stage is the sporophyte, and they're named after what they make. So the gametophyte makes the gametes, and the sporophyte makes the spores. Now I'm going to walk you through this first with a diagram, and I'm going to give you kind of the text version of what's happening during the cycle, but then I'm also going to draw it on the board in diagrammatic form and walk you through it step by step with color-coded markers so that you can make sure that you understand exactly how this is working and find an easier way to walk yourself through the cycle and make sure you know it. Because I'm going to tell you right now, you need to know this. Backwards, forwards, upside down, you need to be able to draw this by yourself, right? You have to know the alternation generation life cycle. There's so many things about plants that are based off of this. So we're going to know this. We're going to learn this one in detail. Okay? Now, which one of these stages right here did we not see in the haplontic life cycle?
Sporophyte.
The sporophyte, exactly right. The haplontic life cycle had a multicellular haploid stage. It did not have a multicellular diploid stage. That's important. That's why the sporophyte is one of the shared direct traits of land plants. It's because it's in this life cycle, not in the previous one that we saw. Okay? It is definitive for this group. Okay? So we're going to start here with our gametophyte down here in the bottom. This is the multicellular haploid stage, which is similar to what we see in the haplontic life cycle. And that stage right there is going to form the gametes through the process of mitosis. This is where students oftentimes get mixed up because they usually think about gamete production in terms of humans. And we have diploid cells in our body that undergo meiosis to make our haploid gametes. And we always think meiosis. Meiosis makes gametes. Not in plants. Doesn't do that in plants. Right? This thing down here, this multicellular stage, is haploid. And the gametes are haploid as well, which means that it can't be meiosis that's used to make the gametes. It has to be mitosis. We're going to kind of review what mitosis and meiosis do. Right? Think about these different stages. And we're doing the basics here. We're not going into detail like we see for Biology 140 or anything like that. Right? We've got meiosis. We've got mitosis. Meiosis actually involves two different cell divisions, right? You're going to start off with one cell right here, and that cell is going to divide into two. And then each of those is actually going to divide so that you get a total of 4. So you start with 1, you end up with 4 through the process of meiosis. Second thing to know about meiosis is that it cuts your chromosome number in half. Right? So, for example, if you started with a diploid cell up here, I'll use the abbreviation 2N because that's usually what we use for that, then you would end up with cells down here that are haploid. That's why meiosis can't be involved in this process right here, because it cuts that chromosome number in half. And if you're already starting with haploid, how do you cut that in half? You end up with 0.5 chromosome sets. That doesn't work. Right? But if you go over to mitosis, this one only involves 1 cell division. So you end up with only 2 cells if you started with just 1. And your chromosome number stays the same. Right, so that means if we started off here with a diploid cell, 2A, we'd end up with 2 diploid cells that are essentially copies of the parent that they came from. They're clones. The same thing applies though if you started with a haploid cell. If you started with a haploid cell, the 2 cells that you would make would be haploid. And honestly, it doesn't matter what you start with as long as what you end up with is the same in terms of the chromosome number. Right? So that's what's happening here. We started off with something that was haploid, we went through mitosis, and we ended up with gametes that are haploid. Right? All right, next up, we have our gametes now. I should just let you jump if you don't. Wait till we get the, like, the diagram with all the colors on, and you'll be excited for the picture. I'll even turn the lights on for you for that. Alright, so, once you have your gametes, those gametes are gonna fuse together in the process of fertilization, and you're going to get your first cell, which is the baby plant right here, that's gonna eventually form into the sporophyte that's called the zygote. The zygote is diploid though, because both of those gametes were haploid. When you bring two haploid cells together and fuse them, you're gonna get a diploid cell. It is a single cell though, and remember, land plants are multicellular organisms. So that means that at some point this is gonna start to divide and grow and become multicellular. But it goes through a couple of stages to get there. Okay? When it initially starts to divide, okay, it's going to form into something called an embryo. Here's where the process deviates from what we saw from the haplontic life cycle. In the haplontic life cycle, the zygote was formed. You had gametes fused together, you formed a zygote. But what that zygote did is it immediately underwent meiosis and it made spores all the way over here. Okay? It went through meiosis, so it was a single cell, it formed 4 spores. The haplontic or the haplodiplontic life cycle or alternation of generations life cycle doesn't do that. The zygote delays that meiosis, okay? And the first thing it does is it starts to divide and grow, okay? And it forms into that embryo. That's why, again, the embryo stage is considered a shared derived trait for the land plants as well, because we didn't see that in the haplontic life cycle. We never got to a multicellular stage. This is multicellular. It's not real big, but it's multicellular. Okay, so what is the embryo? Well, it's basically just your immature sporophyte, what's going to eventually become the main plant, at least for most of the plants. It's attached to and surrounded by the gametophyte. In some cases, this is what's essentially providing the food source for it. And if we're talking about seed plants, so inside of a seed, you've got the little embryo there, it's got nutritive tissue around it, that's usually the gametophyte that's providing food for that embryo until it's able to germinate out of the seed and get its own resources through roots and leaves. Right now it doesn't have roots and leaves yet. It's not present in haplontic life cycles, and so this is actually why we call land plants embryophytes, because it's a shared derived trait for them. Embryophytes. Now this can remain dormant, okay, for a certain period of time. It doesn't actually have to continue developing yet into the sporophyte until conditions are more favorable. It's one of the advantages of having that embryo. That's why it's adaptive. We talk about having adaptive advantages and why these shared derived traits are adaptive. This is why it's adaptive. You don't have to germinate, you don't have to develop right away. Wait until conditions are better. That's why seeds can stay dormant for so long. And then this is eventually going to continue to grow and divide through mitosis to mature into our sporophyte. Okay, so let's talk about our sporophyte. It makes what? Spores. Exactly right. But it's not going to make spores over the entire body. Right? It's going to have special organs that are responsible for making spores. Okay? So there's a portion of the sporophyte that differentiates into something called a sporangium, and that's ultimately what makes the spores. Think about it in terms of humans. We don't make gametes throughout our body. We have special organs that make those. Same thing applies here for the spores. Same thing's going to apply for the gametes. They're going to have special organs for doing that. Now, how does it make these? This is where meiosis is going to happen. This is the only place where meiosis is going to happen in the alternation generations life cycle. Everything else is either fertilization or mitosis. So it makes the spores, not the gametes. Once the spores are produced, they're gonna divide by mitosis and develop back into the stage that we started with the gametophytes. Now, I know that's a lot of words, and so you're trying to walk yourself through it with text. Sometimes it can seem confusing and overwhelming. So let's try and find a simpler way for us to remember this cycle. Everybody, right leg. Alright, now if you've been in my 130 class, you have gotten some of this before, right? Because I always draw alternation generations on the board. But I told you when you had 130 with me that there was a stage missing that we don't talk about in 130, because it's a little bit too advanced for that. This class we are, it's gonna be the embryo stage. So if you remember that from 130, we're gonna add an extra stage in here that we didn't have before. Alright. We're basically drawing alternation generations on the board. It doesn't matter where you start the different stages from, just make sure that they're pointing for the right thing that they make. So I'm just gonna put our multicellular stages up first. I'm gonna start with putting the sporophyte on top. And I'm gonna put the gametophyte down here at the bottom. Step number 1, right? Next step, right? We're gonna indicate what each of these stages makes. The sporophyte makes spores, okay? So we're gonna put spores over here, okay? And the gametophyte makes gametes, and there's 2 different types, male gamete, female gamete. What are they? And sperm and egg, exactly right. So we're gonna have, this is gonna be a bisexual gametophyte. We'll get into unisexual later. This is a bisexual gametophyte, so we're gonna make sperm, and it's gonna be able to make the egg. Okay? Now, those 2 gametes will come together in the process of fertilization. They will make the next stage. What's that first cell that's formed after fertilization?
Zygote.
A zygote. That's right. So we make her a zygote, single cell. That is going to start dividing and growing, become multicellular, but it may not continue the process all the way to the top yet because it's going to go through kind of a dormant stage. What was that stage that was kind of the in-between stage? The embryo. Okay, you're right. We got our embryo. That's what I did put in 130. Okay, so you're wondering where that extra step is. That's it right there. Okay. Embryo stage. Okay. Once that does start to develop, okay, after it goes through whatever dormancy stage it's going to go through, it will eventually develop back into our sporophyte. Okay. This is a cycle, so we got to connect the lines here. Only thing left we have to do here is indicate what the spores are going to become. And we've just talked about right here, the spores divide by mitosis and develop into the gametophyte. So all we have to do here for this is draw a line down to there. So far, so good, right? We can handle this, right? This is just memorization. We're just gonna have to memorize this. Right? Walk yourself through it step by step. All right, next thing we're gonna do, I'm gonna get a different colored marker. If you have a different colored pen, I encourage you to use different colors here. We are going to indicate what the ploidy level is for each one of these stages right here. Is it haploid, is it diploid? I'm going to use abbreviations, so I need to define what those are. If I do 2N, that's going to equal diploid. If I use N, that's haploid. Now remember what those mean. Diploid means you've got 2 copies of each of your chromosomes, and haploid means you've just got 1 copy of each of your chromosomes. Okay? This you're just going to have to memorize. But if you can do this, the next step and the next thing we're going to fill in on the chart will become much easier. Okay? So, going through first, let's just look at our multicellular stages, our sporophyte, gametophyte. One of them is diploid, one of them is haploid. Which is which? Diploid's the sporophyte, so we've got 2N for this one, and we've got N for this one. Okay. How about our spores? I'm sorry? Haploid, exactly right. Spores are haploid. Gametes? Same as ours. Haploid. Sperm's gonna be haploid, eggs gonna be haploid. How about our zygote? Diploid, exactly right. You're bringing those 2 together and you're forming something that's diploid. And our embryo. Diploid. Okay, so here's one thing that's nice about this. There are 3 stages that are diploid. There are 3 stages that are haploid. that'll help you remember it, 3 and 3. Okay. All right, next step, we'll use a different color now for this one. We need to put in what processes are involved for each one of these different stages. We're gonna be using meiosis, mitosis, fertilization. Those are our processes. I'm gonna use blue for that. Now, knowing what you now know about what happens to the chromosome number right here, when you do mitosis versus mitosis, this should be pretty easy if you have your numbers right here that you put in your chart. If you're going from diploid to haploid, what process has to be involved? Meiosis, again, you're right. Again, this is the only place that meiosis occurs. If you're going from haploid to haploid, it is mitosis. Essentially what's happening here is you're going from a single cell, you're dividing that and growing, which is what mitosis is used for, to become something that's multicellular. Okay? And then here we're going from haploid to haploid. Mitosis. All right. So to make the gametes, we use mitosis. Tricky question. Haploid to diploid. Fertilization. Good. Diploid. Mitosis. Again, we're just growing and dividing here. And diploid. Mitosis. So the good news is that pretty much everything's mitosis, with the exception of 2 spots. Fertilization, and hopefully everybody can get that one right, and meiosis. So if you can remember where the meiosis goes or where the fertilization goes, everything else is gonna be mitosis. I'll put that in there. Now, the only thing we can add in here, unfortunately I don't have my red marker with me, so I'll have to use black again, is we can indicate whether each of these stages is a single cell or if it's multicellular. So I'll use multi and multicellular. And I'll use uni or unicellular. Okay. Sporophyte. Multicellular, good. Spores? Unicellular, good. Gametophyte. Multi. Gametes? Uni. Zygote? Uni. It's a single cell, that's right. Embryo? Multi. And sporophyte, we already got multi. Okay? So again, if we look at our numbers here, we have 1, 2, 3 stages that are multicellular, and we have 1, 2, 3 stages that are unicellular. 3 and 3, it's always 3 and 3. It's not always the same 3 and 3, but it's gonna be 3 and 3. 3 that are diploid, 3 that are haploid, 3 that are unicellular, 3 that are multicellular, right? To help you with your numbers to get that. That's the alternation generations life cycle, that's it, right? You feel comfortable we can do that. Can you walk yourselves through this? There's some memorization involved, yes, but there's also some logic involved as well if you can understand how the process of mitosis and meiosis work, right? You should be able to do this on the test, right? I'm gonna tell you right now, there's gonna be a question that says draw alternation generations. Tell me the stages, tell me where the processes occur, tell me where the diploid or haploid, you have to do all of that, right? This is the answer to the question right here, right? Make sure you know that. All right, now why are we learning this so much? And why is this sporophyte stage so adaptive in land plants? Well, it's got several advantages. If you can have a multicellular diploid stage as a stage in your life cycle, as opposed to just a multicellular haploid stage, you've got some big advantages. Okay? One, you're gonna be able to make more spores, because that's what that stage does, is it makes spores. Think about what happened in the haplontic life cycle. Okay? You had your gametes fused together and you got a zygote. That's the only diploid stage that's in this life cycle, is this single cell, one cell, and that's it. Okay? If it undergoes meiosis, how many spores is it capable of making? Meiosis divides twice and you get how many cells? 4. That's all they can make, 4. This single cell right here can only make 4 spores, and that's it. That's their life cycle, right? But if we go into alternation of generations and you have a sporophyte, it is multicellular. There are multiple cells in here called sporocytes that are undergoing meiosis, and each one of them is making 4 spores. So you have this exponentially higher number of spores that are produced by having a multicellular diploid stage than by having just this unicellular one right here, right? So that's offspring. That's more offspring you can make. That's an advantage. Second, you have 2 copies of each gene. But just because you have 2 copies of each gene doesn't necessarily mean that you have the same allele on each one of those chromosomes. Right? So you're getting some of this from your mother, you're getting some from your father. We're going to harken back to our genetics here. Hopefully everybody's not having bad memories about this. Okay? But everybody remembers heterozygous, homozygous, those different characteristics related to whether or not you have the same allele on both of the chromosomes or different alleles. Right? So if you have a recessive allele, right, something that's masked if a dominant allele is present, and it's deleterious, which means that it actually decreases your fitness, in some cases it kills you to have that, you don't necessarily want that allele to be expressed. Right? Everybody got the alternation of generations here? Do I need to step out some pictures? Okay, so deleterious is any mutation or allele or trait that, if it's expressed, reduces an individual's fitness, their ability to survive and reproduce. Okay, so let's use an example here. Let's say we have 2 alleles here, big D and little d. This is the dominant one, so if it's present, it'll be expressed. This is the recessive one, it's only expressed if it's the only one that's there. If you have the dominant allele, you're okay. You have an okay phenotype. You're going to be able to survive and reproduce. You're going to have a normal lifespan. Of course, if you eat, that's a different story, but you're not going to die as a result of this allele right here. Your fitness is not being reduced by this. This one's recessive. I'll add a word in here. Okay. This is a recessive deleterious allele. It reduces the fitness of the individual if it's expressed. Right? Now, if you have, or if you are a haploid individual, you only have 1 copy of each of your chromosomes and 1 copy of this gene. You can either have one or the other allele. You can't have both. Right? That means your phenotype is either a D, in which case you're going to be okay, or it's little d, in which case you're not. What that means is that you have a 50-50 chance of being okay. And a 50% chance of having reduced fitness. Now, look at our diploid options. That means you have 2 copies of your chromosome, of that gene. You can have the same allele on both of those, or you can have different alleles, right? So if you have the same allele on both of them, these are your 2 possibilities. You could be homozygous dominant, in which case you're okay. You could be homozygous recessive, in which case you're not. Or you could be heterozygous. Okay or not? They're okay, right, you're right, because it's gonna get masked. So this means if you're diploid, you only have a 33% chance of having reduced fitness. That's an advantage. Is it all coming back to us? Bad memories? Okay, so if you have a reduced chance of having reduced fitness, that's an advantage. Okay? You could potentially mask a bad recessive allele that you might have and still be okay. Right? You're still gonna have that allele, and yes, it can still be passed on, but you're okay in terms of your phenotype because you've masked it with this allele. You can't do that if you're haploid. Right? So having that diploid stage could be very valuable for that reason. That's one of the adaptive advantages of that. Okay? All right, we're at 1:49. I think we're gonna stop here for today because we're doing stopping place. We'll get into the people.